Right now, inside your body, there are cells carrying your mother's DNA, detectable under a microscope, identifiable by her genome, engrafted into your organs and dividing in your tissues. If your mother is still alive, her cells are in your body, and your cells are in hers. Two bodies, each containing the other, each carrying a permanent cellular population that originated in the other. If your mother has died, whether last year or decades ago, her cells are still in your body. The cells do not know she has died. They are not connected to her by any signal that her death would interrupt. They engrafted during pregnancy. They became part of your tissue. They remain. The discovery that made this known arrived in 1996. A pathologist named Diana Bianchi at Tufts University was studying fetal cells in maternal blood, cells from the baby that cross the placenta during pregnancy and enter the mother's circulation. The research question was straightforward. How long do these cells persist after delivery? She expected weeks, perhaps months. The immune system clears foreign cells. That is what the immune system does. She did not expect what she found next. Fetal cells were present in maternal blood samples taken from women whose pregnancies had ended decades earlier. 27 years after giving birth, a mother's blood still contained her child's cells, alive, dividing, carrying the child's DNA in the mother's body. The cells had not been cleared by the immune system. They had not degraded. They had not been attacked as foreign. They had engrafted, embedded in her organs, her tissues, her bone marrow, and become a permanent part of her body. The immune system that should have destroyed them had instead accepted them. And then the reverse was confirmed. Maternal cells cross into the fetus during pregnancy and persist in the child's body into adulthood. The cells Bianchi found were not a one-way phenomenon. The trafficking is bidirectional. Trafficking begins during pregnancy. Fetal cells, carrying the combined genome of both parents, genetically distinct from the mother, enter the maternal bloodstream through breaks in the placental barrier. Maternal cells make the crossing in the other direction. The mother's cells, carrying her genome, enter the developing child's body through the placenta. The exchange is bidirectional, continuous across pregnancy, and the cells that survive the immune system's initial response do not leave. They engraft. They become permanent residents in a body that is not genetically their own. The term is microchimerism. Chimera, from the Greek mythological creature composed of parts from different animals. Two genomes in one body. Bianchi and colleagues published in the Proceedings of the National Academy of Sciences that male fetal cells, identified by the presence of Y chromosome sequences, were detectable in maternal blood up to 27 years after the birth of a son. The Y chromosome was the detection method, a genetic marker the mother's own cells could not carry, making the fetal cells unambiguously identifiable in maternal tissue. Subsequent studies found fetal cells engrafted in maternal liver, thyroid, brain, heart, kidney, and skin, not clustered in one organ, but distributed throughout the body, as though the cells had entered the bloodstream and migrated to wherever the tissue would accept them. These cells were not inert passengers sitting in tissue doing nothing. In the thyroid, fetal cells had differentiated into functioning thyroid cells. In the liver, they were found participating in tissue repair. In the brain, they were integrated into neural tissue, functioning neurons carrying the child's genome in the mother's brain. Her body does not merely tolerate these foreign cells. In some cases, it appears to use them. Studies of women with liver damage found higher concentrations of fetal microchimeric cells at sites of injury. The cells had migrated to damaged tissue and contributed to repair. The relationship is not uniformly benign. Other studies associate fetal microchimeric cells with autoimmune conditions, including thyroiditis and scleroderma, suggesting the cells may participate in both repair and disease depending on context. The cells are present. Whether they are consistently beneficial is not resolved. What is established, the child's cells, decades after birth, are active participants in the mother's tissue biology, repairing in some cases, potentially contributing to pathology in others, but present and functioning either way. Reverse direction. Your mother's cells in your body operates through identical biology. Maloney and colleagues published in the Journal of Clinical Investigation that maternal cells were detectable in the blood of healthy adults decades after birth. Maternal cells have been found in the child's liver, lung, thymus, skin, and lymph nodes. Thymus finding is the one that matters most for what follows. The thymus is the organ that trains your T cells to distinguish self from non-self, the organ that teaches your immune system what belongs in your body and what does not. Maternal microchimeric cells have been found among the thymic epithelial cells that present self-antigens during T-cell education. The implication is extraordinary. Your immune system may have been partially educated by your mother's cells. The definition of self that your immune system learned during development may include molecular signatures from your mother's genome. She is not only inside you, she is part of how your body defines what belongs. Think about that for a moment. The immune system that protects your body, that identifies every virus, every bacterium, every foreign protein as not self, was partly trained by cells carrying your mother's DNA. The boundary between self and other, which the immune system enforces at every moment of your life, was drawn with her cells participating in the drawing. The line between you and her was never as sharp as biology textbooks suggest. At the cellular level, the line does not exist. That finding, your immune system's definition of self, including molecular signatures from your mother's genome, the boundary between you and her being an artifact of textbook simplification rather than biological reality, is the finding that made the self-non-self distinction look like a simplification rather than a boundary. Because it means the most fundamental distinction biology makes, self versus non-self, was co-authored. She is not a foreign presence your body tolerates. She is part of the definition your body uses to decide what belongs. Cells are only the first layer, because your mother's physical presence in your body extends beyond the cells she left behind, into the chemical modification of your DNA itself. During pregnancy, the fetal epigenome, the chemical modification layer sitting on top of DNA that controls which genes are expressed and which remain silent, is actively shaped by the maternal environment. Cortisol levels in the mother's blood cross the placenta and influence methylation patterns on the fetal genome. Nutritional availability determines methylation of metabolic genes. Inflammatory markers shape immune gene expression. What the mother experienced during pregnancy, her stress, her nutrition, her health, wrote chemical instructions on your DNA that persist for your lifetime. Dutch hunger winter provides the clearest evidence. During the winter of 1944-45, a Nazi blockade cut food supplies to the Western Netherlands. Pregnant women experienced severe caloric restriction. Hagemans and colleagues published in Nature Genetics that children conceived during the famine had measurably different methylation patterns on the IGF-2 gene, a growth-regulating gene, compared to their siblings conceived before or after the famine. These methylation differences persisted 60 years later. The famine the mother experienced in 1944 was still chemically present on her child's DNA in 2008. Her experience during 9 months of pregnancy had written a chemical mark on the child's genome that 6 decades of subsequent life did not erase. What she lived through is written on what you carry. The physical record of her experience persists in your cells in a form that influences your gene expression today. The epigenetic marks are not memories. They are chemical modifications, methyl groups attached to specific cytosine bases on specific genes, placed during the months you were inside her, governing the expression of those genes for the remainder of your life. Deeper finding, though it requires careful qualification. The grandchildren of famine-exposed women showed metabolic differences consistent with the grandmother's experience. Whether this represents true transgenerational epigenetic transmission or multi-generational in utero exposure is actively debated. The grandchild's precursor cells were already present in the mother's ovaries during the grandmother's pregnancy, meaning the exposure may have been direct rather than inherited through an epigenetic mechanism that crossed a generation without physical contact. The distinction matters. What is established? The metabolic differences in the grandchildren are measurable and consistent with the grandmother's experience. whether the chemistry crossed one generation or two, whether the mark was placed directly on the grandchild's precursor cells or transmitted through an inheritance mechanism, the physical record of what she lived through persists in bodies she may never have met. Now, before birth, before cells, before chemistry, because the first connection between you and your mother was not cellular, it was acoustic. Human fetus begins hearing at approximately 18 weeks of gestation. The cochlea is functional. The auditory cortex begins processing acoustic input. The dominant sound in the uterine environment, louder than the heartbeat, louder than digestive sounds, louder than any external voice, is the mother's voice. Not transmitted through air, transmitted through bone conduction and fluid, from her larynx, through the tissues of her body into the uterine wall, and through the amniotic fluid to the fetal ear. The father's voice and external sounds reach the fetus through air, to tissue to fluid transmission, attenuated and filtered by every layer they pass through. The mother's voice arrives through direct tissue conduction, louder, richer, and less filtered than any other sound in the fetal acoustic environment. De Casper and Pfeiffer published in Science in 1980 a finding that changed developmental neuroscience. Newborns, tested within hours of birth, modified their sucking patterns specifically to activate a recording of their mother's voice over a stranger's voice. They had never seen her face. They had never been held outside the womb. They recognized the voice they had heard through fluid and bone for 4 months. The auditory cortex had encoded her vocal signature, her pitch, her rhythm, her phonemic patterns during fetal development. De Casper and Spence followed in 1986 with a finding even more specific. Mothers who read a particular passage aloud repeatedly during the last 6 weeks of pregnancy produced newborns who preferentially responded to that passage over an unfamiliar one, even when read by a stranger. The fetus had learned not only the mother's voice, but the specific prosodic patterns of a text she repeated. Her speech patterns were encoded in your auditory cortex before you were born. The voice you recognize as hers was the first sound your brain ever processed, and the encoding is older and deeper than any postnatal memory you carry. If your mother is still alive and you hear her voice on the phone, the immediate recognition, the emotional response that arrives before the first sentence is complete, that is not adult pattern recognition. That is an auditory encoding laid down 4 months before your birth, through fluid and bone, in an auditory cortex that had never processed any other voice. Her voice is not one voice among many that you learn to recognize. It is the foundational voice, the acoustic template against which every subsequent voice was measured. The encoding predates language. It predates vision. It predates every other sensory experience you have ever had. You see, and the auditory bond was only one channel of the physiological coupling between your body and hers. Because during the first months of your life, her body was not merely near yours, it was regulating yours. Ruth Feldman at Bar-Ilan University has spent 2 decades measuring the physiological coupling between mothers and infants. Using dual heart rate monitors during face-to-face interaction, she demonstrated that mother and infant heart rates synchronize. The 2 hearts begin beating in phase within seconds of mutual gaze. The mechanism is autonomic. Mutual gaze activates the ventral vagal complex in both mother and infant, producing coordinated parasympathetic output that entrains cardiac rhythm across the visual autonomic feedback loop. 2 separate hearts, beating in phase, driven by eye contact, measurable entrainment. The cardiac cycles aligning within seconds and maintaining alignment for the duration of the mutual gaze. Synchronization extends beyond heart rate. Feldman's laboratory measured simultaneous cortisol levels, oxytocin levels, skin conductance, and respiratory rate, all synchronized during sustained contact. 2 separate bodies, 2 separate nervous systems, 2 separate endocrine systems running in phase. Her calm calmed you. Your distress activated her. The coupling was bidirectional and physiological, not emotional resonance in the metaphorical sense, but measurable entrainment of autonomous biological systems across 2 bodies that were, in every physiological sense, operating as one regulatory unit. Evolutionary logic is straightforward. The infant cannot regulate its own autonomic state. Its thermoregulation is immature. Without the mother's body heat, the infant's core temperature drops. Its cortisol system is uncalibrated. Without the mother's cortisol rhythm as a template, the infant's stress response oscillates without a stable baseline. Its vagal tone is developing. Without the mother's parasympathetic output as an entrainment signal, the infant's heart rate variability remains erratic. The mother's body serves as an external regulator. Her physiological state is the template the infant's developing systems calibrate to. Hoffer at Columbia University called these hidden regulators. The mother's heartbeat, warmth, scent, and voice are regulatory inputs that the infant's nervous system depends on for calibration. They are hidden because they operate below awareness. Neither the mother nor the infant knows that her heartbeat is setting his autonomic baseline, that her cortisol rhythm is calibrating his stress response, that her body temperature is regulating his thermoregulation setpoint. The regulation is invisible and continuous. Separation from these inputs produces measurable physiological dysregulation. Heart rate variability decreases, cortisol spikes, sleep architecture fragments. The infant's body does not merely prefer the mother's proximity. It requires it as a regulatory input, the way a thermostat requires a temperature signal. The settings established during this calibration period persist into adulthood. The resting heart rate variability you carry at 65 was partly calibrated by the heart that beat next to yours at 6 months. The cortisol rhythm you carry, the daily rise and fall of your stress hormone, was partly set by the rhythm her body modeled during the months when your developing nervous system was using her body as its reference signal. These are not metaphors. They are calibration settings, set during a developmental window, persistent across a lifetime. Breast milk added another layer, an immunological transfer that wrote her pathogen history into your defenses. Breast milk contains secretory immunoglobulin, A, SIA antibodies produced by the mother's mucosal immune system in response to every pathogen she encountered and survived across her entire life before your birth. Each SIAA molecule is specific to a particular pathogen surface protein, a lock that fits one key, designed by her immune system after it defeated that specific pathogen. Every infection she fought, every cold she recovered from, every pathogen her immune system learned to recognize, encoded in the diversity of SIGA molecules in her milk. When you ingested that milk, the SIGA coated your gut and respiratory mucosal surfaces, providing passive protection against pathogens you had never encountered, but she had already defeated. Transfer was not random. The enter-o-mammary pathway operates as a real-time dispatch system. Pathogens the mother encounters in her gut stimulate her intestinal immune cells, which migrate to the mammary gland and produce SIGA locally, which enters the milk and reaches the infant's gut within 24 to 48 hours. If she was exposed to a new pathogen on Monday, her milk contained antibodies against it by Wednesday. The milk was an updating immune report, a real-time summary of the pathogen environment you were about to face, authored by a body that had already survived it. Your adult immune system is not carrying her antibodies. The SIGA molecules were temporary, gone within weeks of weaning, cleared from your mucosal surfaces once breastfeeding ended. The protection was transient, but the survival it enabled was permanent. The infections her antibodies prevented during the months when your own immune system was immature. The pathogens her SIGA neutralized before they could establish in your gut, your respiratory tract, your developing body, shape which diseases you survived and which you never encountered. Her immune autobiography did not persist as molecules. It persisted as the fact that you lived through infancy with a borrowed immune system covering the gap between birth and the maturation of your own defenses. The antibodies are decades gone. The survival they enabled is the body you carry. Well, and then there is the scent. Because the olfactory recognition between mother and child operates through a pathway faster than any other sense, and the molecular basis of the recognition is immunological. Newborns identify their mother by olfactory signature within hours of birth. Porter and Winberg published that neonates preferentially orient toward breast pads worn by their own mothers versus other lactating women tested within the first week of life. The recognition is based on volatile compounds produced by the Montgomery glands on the areola, a sebaceous secretion containing the mother's MHC-linked volatile profile. The MHC genes, the genes that determine tissue compatibility for organ transplants, produce volatile organic compounds unique to each individual. Your mother's scent was a molecular identity card, genetically determined, immunologically specific. No two mothers smell identical to the newborn. The recognition is precise enough to distinguish one mother from every other lactating woman in the room, through chemistry alone, before the eyes can focus on a face. Olfactory, limbic pathway, the direct connection from olfactory bulb to hippocampus and amygdala, the only sensory pathway that bypasses the thalamic relay, means that her scent triggers memory and emotion faster and more intensely than any other sensory modality. A specific scent, her perfume, the smell of her cooking, the particular character of the home she kept, produces autobiographical memory retrieval and emotional response through the fastest sensory route your brain possesses. The nose recognizes her before the mind does. For the person over 65 whose mother has been dead for decades, a specific scent can produce full emotional transport before conscious identification of what is being smelled. The limbic system responding to a molecular signature it encoded in infancy, before language, before explicit memory, before the conscious mind had any say in what was being recorded. The sudden emotion that arrives when you walk into a kitchen that smells like hers, or catch a trace of the perfume she wore, or encounter the specific combination of soap and fabric and cooking that characterized her home. That emotion is not nostalgia. It is molecular recognition. Your olfactory system is detecting a chemical signature it encoded before you could speak, and it is activating memory and emotion through a pathway that does not pass through conscious processing. She arrives in your awareness before you know why. And then, the brain she built. The final layer, and the most architecturally permanent. The human brain at birth contains approximately 100 billion neurons. The connections between them, the synapses, are massively overproduced during the first 2 years and then selectively pruned based on experience. At peak density, around age 2, the infant brain contains approximately twice as many synapses as the adult brain. Activity-dependent synaptic elimination preserves connections that are repeatedly activated and eliminates connections that are not. The experiences of the first 12 to 24 months determine which pathways survive. Maternal responsiveness, the consistency, timing, and quality of the mother's response to the infant signals, is the primary input driving this synaptic selection. Luby and colleagues published in the Proceedings of the National Academy of Sciences that children whose mothers were measured as more nurturing during early childhood had significantly larger hippocampal volumes at age 13, the hippocampus being the structure critical for memory, spatial reasoning, and stress regulation. The association is consistent, significant, and specific to the early window. Later nurturing, however attentive, however loving, did not produce equivalent hippocampal growth. Whether the nurturing itself is the causal variable or a marker of broader developmental conditions, genetics, socioeconomic stability, maternal mental health, has not been fully isolated. But the specificity of the timing window and the absence of equivalent effects from later nurturing suggest the early interaction pattern is doing substantial independent work. The brain architecture was physically associated with her behavior during a specific developmental period that neither you nor she chose. The window's specificity is what makes the finding consequential for the person over 65. The brain you carry now was architecturally determined during a period you cannot remember. The first 12 to 24 months, before explicit memory formed, before language allowed experience to be encoded in words, you have no memory of the period that shaped the brain you used to remember everything else. She was building the architecture during a window you will never recall. The hippocampus she shaped is the structure you use to retrieve every memory you have, including every memory of her. She built the organ that remembers her. Consider what that means. The brain you carry at 65, the hippocampus that stores your memories, the amygdala that generates your emotional responses, The prefrontal cortex that governs your decisions was physically sculpted by the interaction pattern between you and her during the first years of your life. The synaptic architecture that makes you think the way you think, remember the way you remember, and respond to threat. The way you respond was selected by her presence, her responsiveness, her voice, her touch. The brain is not self-constructed, it is co-constructed. And the co-constructor was her. Every thought you have, every memory you retrieve, every decision you make, every emotional response you produce travels through synaptic pathways that her behavior selected for survival during the pruning window. The pathways she activated by responding to you were preserved. The pathways she did not activate were eliminated. The neural architecture you use today is the architecture her responsiveness chose. Through the physics of activity-dependent synaptic selection, the pathways that fire together survive together, and the firing was driven by her. That inventory, cells carrying her genome engrafted in your organs, epigenetic marks she placed on your DNA during pregnancy, her voice encoded in your auditory cortex before birth, your autonomic calibration set by her heart rate and cortisol rhythm, her scent encoded in the fastest pathway your brain possesses, and her responsiveness sculpted into the synaptic architecture you still think with, is the inventory that will not resolve in the developmental biology literature. 6 systems that persist physically in your body today. A 7th, her immune protection through breast milk, that persisted long enough to carry you through the months when your own defenses could not. Each independently documented, each independently measured, and all operating from one origin, the months and years when your body and hers were not fully separate. Now, what persists after she dies? Because every system described above continues operating in your body regardless of whether she is alive. When she dies, whether that happened last year or 40 years ago, the cultural framing says she is gone. The physics says otherwise. Her cells remain in your body, carrying her DNA, engrafted in your organs. Cells that were placed during the months you were inside her and have been dividing in your tissues ever since. Her epigenetic marks remain on your genome, methylation patterns placed during pregnancy that govern gene expression in your cells today, 60 or 70 years after they were written. Her voice remains encoded in your auditory cortex. The first sound your brain ever processed, laid down 4 months before birth, retrievable through memory for the rest of your life. Her heart rate calibrated the autonomic settings you still carry. The resting heart rate variability set during the months when your developing nervous system used her body as its regulatory template. Her responsiveness shaped the synaptic architecture you still think with. The hippocampal volume, the amygdala calibration, the prefrontal connectivity selected by her behavior during the first years of your life. Her scent remains encoded in the olfactory limbic pathway, retrievable by a single molecule in the air, producing full emotional recall through the brain's fastest sensory route, before conscious thought has identified what triggered it. And her immune protection. Temporary in its molecules, permanent in what it preserved, carried you through the window when your own defenses could not. Mother who has been dead for 20 years is present in her child's cells, DNA, auditory cortex, immune system, autonomic calibration, brain architecture, and olfactory memory. As a physical substrate, measurable, detectable, distributed across 7 biological systems that do not require her to be alive in order to continue carrying her. The grief that does not fully resolve, the sense that she is somehow still present, still carried, still part of the body, is the body reporting an accurate inventory of what it contains. She is still present. The body is not confused. The body is correct. Diana Bianchi found the cells in 1996. 27 years after birth, the child's cells were still inside the mother's body, alive, dividing, engrafted in organs the child had never touched. The mother's cells were still inside the child, integrated into tissues, participating in immune education, carrying her genome in a body that defines itself partly through her molecular signatures. 2 bodies, permanently containing each other at the cellular level, from the moment the placenta formed until the last cell divides. The placenta dissolved within hours of birth. The cellular exchange it enabled has lasted decades. The temporary organ created a permanent condition. Next time you think of her, whether she is alive or whether she has been gone for decades, know that the thought is not arriving in a body that has lost her. It is arriving in a body that contains her, her cells, her chemical marks on your DNA, her voice in your auditory cortex, her responsiveness in your synaptic architecture, her scent in the fastest pathway your brain possesses. She is not remembered, she is carried. The physics does not use the past tense.